Hello and welcome back to Survey of Biology, and this is the Mendelian Genetics, Unit 10, and we're on Section 4. So this is going to be the last of the sections, and here we're going to try to introduce something called dihybrid crosses. So if you remember the last section, we talked about Punnett squares and how we could look at one trait at a time in monohybrid crosses, and what we're doing now is we're going to look at two traits at the same time. So the case that we'll look at here is, what if we take a gene for tall versus short or dwarf pea plants, so big T versus little t, and then we also look at the same time purple versus white flowers for those pea plants, big B versus little p. So if we take an individual that's completely heterozygous for both traits, big T, big P, little p, and we form gametes. So if you remember, during gametes, according to law of segregation, each of those alleles, the big T and little t, must be separated. So they can only get a big T or a little t. They can't get both. And the same is true of the P gene. They can either get a big P or a little p. They can't get both. So based on that, we can form 4 different gametes between those two traits. And if we look here between the yellow and the sphere, which is the dominant traits for a pea seed, we can see again big S, big Y, little y. They can be segregated into 4 different gametes, but the big thing to remember is that we have to have one of each gene in there. We can't have big S, big S. That would not be allowed. We can't have big Y, little y in a gamete because the genes for the same trait have to be segregated and separated out so that they're only inheriting one gene from each parent from the gametes. So if we look at a Punnett square here, here's one for yellow round, which is the dominant seed shape, versus green wrinkled, which is the recessive. And if you see, if we take the heterozygous on both sides and we have big Y, little y, big Y, little r, we can have 4 different gametes that are formed on each side. So we have a 4x4 Punnett square instead of the 2x2 that we were looking at in the last section. So as we look at these, you can see now there are 16 boxes. So instead of this relatively simple 3-to-1 or 1-to-2-to-1 ratios that we see with a monohybrid, what you need to remember is for a dihybrid cross, we're always going to get this characteristic 9-to-3 to 3-to-1 ratio if both parents are completely heterozygous. And what that means is that 9 of the individuals, 1, 2, 3, 4, 5, 6, 7, 8, 9, will be completely dominant for both traits. 3 of the individuals will be dominant for one trait but not the other. In this case, they'll be round but the recessive green. Another 3 will be dominant for yellow but recessive for wrinkled. And one of them, right down here, will be the recessive for both traits. So 9-to-3 to 3-to-1. And we can look at the same thing in my own writing, if you can make this out. And I'm not going to get to the point where I want you to do out a full Punnett square, but again, just look, if we do a completely heterozygous dihybrid cross for, in this case, traits A and B, we should get 9 individuals circled in black that are going to be the double dominant, 3 individuals in pink that are dominant for A but recessive for B, 3 individuals in the dark green that are recessive for A but dominant for B, and then 1 individual way down here in the corner that's going to be the double recessive. It's recessive for both A and B. So now what I want to try to do quickly is show you a method that will make it a little bit easier and quicker to solve the probabilities for dihybrid crosses. So let's look at this first one. So here we're going to cross 2 individuals, 1 and 2. The first individual is completely heterozygous for both A and B. The second individual is homozygous dominant for A and heterozygous for B. So they want to know, if we cross those 2 individuals, what's the probability that we'll produce offspring that are completely heterozygous? Well, what we need to be able to do is look at each trait separately. So we're going to do 2 individual Punnett squares, one for A and one for B. So for the A gene, one parent is heterozygous, so it can give either a dominant A or a recessive A. The other parent is homozygous dominant, so they're just going to give a big A. So as we can see from the yellow blocks in the Punnett square, there is a 50-50 or 1/2 chance that they will be heterozygous for the A gene. Now let's forget about A for a second. Let's look at B. Both individuals are heterozygous for B, so it can be big B or little b. big B or little b. And again, if we look at the yellow box, half of the 4 offspring, or 1/2, will be completely heterozygous. Now, using the rule of multiplication that we learned before, multiply the numerators, multiply the denominators, there is a 1 in 4, 25% probability that the individuals will be completely heterozygous for both traits. So now let's try to apply that to an example. So here's an example where we've got 2 genes, T and Q, and both parents are completely heterozygous for those traits, and the question is, how many individuals would have both dominant traits? So what we need to do is look at a Punnett square for each one individually. So let's look at T. So if we set up a Punnett square for T, one parent can provide a big T or a little t. The other parent can provide the same. So now if we fill in, what percentage of the offspring will provide a dominant trait? Well, there are 3 out of the 4 offspring that will provide that. Now let's look at the Q. And it's going to be the same situation for the Q if we set up a Punnett square, because both parents are heterozygous. We have 3 out of 4 individuals that will express the dominant trait. Oops, sorry about that. My pen is going a little bit haywire here. But it'll be 3 out of 4. So, what percentage will express the dominant trait? Well, 3/4 times 3/4 gives us 9/16. That's the proportion of individuals from this cross that will express both dominant traits. So now let's look at one more example here. Again, we're going to use T and Q, but this time both parents aren't going to be heterozygous. My pen again, sorry, I apologize for that. Let's see if I can get this cleaned up. So what is the chance that both are going to be dominant? Well, let's do our Punnett square for T. One parent is big T, big T. One parent is big T, little t. So we fill in our Punnett square quickly. And for the T gene, what's the chance that the offspring are going to be dominant? Well, if we look at this, it's going to be 100%. All of them are going to be dominant for T. Let's do the same thing for Q. And one parent is going to be heterozygous. One parent is homozygous recessive. Now, what percentage of these will be dominant? Well, it's 2 out of 4, or 1/2. And then we can do the same thing on the bottom. What's the chance that both are going to be heterozygous? Well, if we look at the Punnett square, I'm just going to pull the Punnett square down from here. What are the chances that they're going to be heterozygous? Well, half of them are. Multiply, we can do the same thing here. We pull the Punnett square down. What are the chances that they're going to be heterozygous? Again, half. So now we can apply our rule of multiplication, and in the top one, it's just going to be 1 times 1/2 equals 1/2, and below 1/2 times 1/2 equals 1/4. So if we look at it, we should have these proportions. So half of the offspring will be the dominant for both. 1/4 will be heterozygous for both. So hopefully if you can take your time and break these down and look at each gene separately, you'll be able to use the rules of probability to do dihybrid crosses in a much quicker and easier manner. So again, thank you for your time, and we will talk to you soon.